Today's topic is enzyme catalysis. Already we discussed the 7 different types of catalysis reactions. This is the last one and very important, enzyme catalysis. Enzymes are complex nitrogenous organic compounds which are produced by living plants and animals. Numerous reactions that occur in the bodies of animals and plants to maintain the life process catalyzed by enzymes. Characteristics of enzyme catalysis. Enzymes are highly efficient, highly specific in nature, highly active under optimum temperature, highly active under optimum pH. Because of these 4 reasons, enzymes are used for catalysis. Examples for catalysis, enzyme catalysis. What are the enzymes utilized in biochemical reactions as a catalyst? Here, invertase enzyme utilized as a catalyst to convert yeast. It is available from yeast, this invertase enzyme, to convert sucrose to glucose and fructose. Sucrose C12H20 when converted into glucose and as well as fructose, we need one enzyme, that is invertase, which is available from yeast. Zymase enzyme convert glucose to ethyl alcohol. The zymase available, the source is yeast. Diastase enzyme converts starch into maltose when malt is the source of diastase. Maltase, yeast, is the source of maltase which converts maltose into glucose. The same way urease is one of the enzyme which converts urea into ammonia and carbon dioxide. This urease is available in soya bean. Pepsin is one of the enzyme which released in our human being stomachs, which is also used as a catalyst to convert proteins into amino acids. Lactobacilli enzyme converts milk into curd. This lactobacilli enzyme is available in source is milk. Mechanism of enzyme catalysis. The enzyme catalyzed reactions may be considered to proceed in 2 steps. Step 1. Binding of enzyme to substrate to form an activated complex. Binding of enzyme and as well as substrate both combine to form an activated complex first. So E stands for here enzyme plus substrate, it means reactant, you will get a activated complex. Step 2. Decomposition of the activated complex to form product. First one is formation of activated complex by the combination of enzyme and substrate, that is reactant, and that activated complex again decomposed into the products. So see here 2 equations are showing in this. E stands for enzyme. This enzyme reacts with substrate. You will get an activated complex E and S both togetherly, which can represent like this. This is the activated complex. And after this decomposition, what you are getting? 2 components you are getting. One is enzyme as it is, and another one is the product. Another one is a product. See here the diagrammatical representation. This is enzyme. This part is enzyme. This is substrate. It means reactant. Both combined together here, enzyme plus substrate, you are getting the activated complex. This one, activated complex. Step 1. Then activated complex in the presence of catalyst. What will happen? Enzyme product complex. This is the complex, activated complex. And after that, activated complex undergoes decomposition. Decomposition. It means divide into, see here, enzyme, these are the products. So this is the enzyme catalysis mechanism in two steps: formation of activated complex, enzyme substrate complex, enzyme product complex. The product will be formed. This will be reacts with this one. You will get the new product here, and then after decomposition you are getting the new products. It means complete yellow color changed into two different colors. These are the two new products are formed in the presence of catalyst. Example, enzyme catalysis. Enzyme, it is a biochemical catalyst, that is urease. This NH2CO, NH2 urea, in presence of urease, it is changed into ammonia and carbon dioxide gas. This is about the mechanism of enzyme catalysis.